Hello, how are you? Today, inshallah, we will start with chapter 12, the DNA chapter. The first lesson of the DNA chapter is identifying the substance of gene. What do you mean by identifying the substance of gene? We need to study the chemical nature of the genes or the genetic material. When we know what is the chemical nature of the gene, we can understand how the gene will affect the functions of the body. For my objective today, I need to identify the genetic material. So we need to identify the genetic material. Again, we need to discover the chemical nature of the gene, and at the same time, if we know the chemical nature of the gene, we will understand how the genes control the inherited characteristics of living things. The first experiments were done by Griffith, and this experiment is known to be transformation or bacterial transformation experiment or a Griffith experiment. So in the beginning, Griffith isolated two strains that's known to be S strain or disease-causing bacteria and R strain harmless bacteria. Two species, two strains of the bacteria, okay? Two strains of the bacteria in the same species, okay? The first one, S strain, caused pneumonia, bacteria or diseases caused bacteria, and at the same time the R strain, it's the harmless bacteria, okay? So the S strain will cause pneumonia while R strain is harmless bacteria. As you can see here, a normal alveoli in the lung is known to be open, free, and the pneumonia or the alveoli for the patients that has pneumonia known to be filled with the mucus, okay? The experiments were done in many stages. The first stage, Griffith injected mice with disease-causing bacteria. And once he injected the mice with S strain, which is disease-causing bacteria, the mouse died because of pneumonia. And the other part, he in the same time injected mice with R strain, okay, and after that the mouse still alive. Then he tried to check what's the main cause of the death of the mouse, okay? The main cause of the death of the mouse, is it the pneumonia itself or the toxin itself? So he tried to change the type of the experiment. He heated the S strain, okay, and after he heated the S strain, he injected the heated S strain inside the bacteria, inside the mice, until then the mice survived. So the next part of the experiments were done by Griffith by killing the bacteria by heating. So he heat the bacteria until it's killed, and then he injected the bacteria into a mouse, and after that the mouse is still alive. So after this part of the experiment, he concluded that the bacterial toxins is not the cause of the death to the mouse, because already the bacteria is dead, but the toxin is found till now. So when he injected the heated killed bacteria inside the mouse still alive. So the main cause for the death is not the toxin, okay? After this part. The other part, or the third part, he make a mixture between heat killed S strain and live R strain. So he make a mixture between heat killed S strain and R strain, okay? And after that, he injected the mixture inside the mouse. After a while, the mouse dies because of the pneumonia. And after an autopsy, he took a specimen, and he already grew the specimen into the plate, and he surprisingly found that the bacteria that already found in the plate is S strain. So what's the main cause? That the dead S strain here and the R strain here changed into S strain here, or lived S strain. So he concluded that the R strain is changed into S strain. How that's happened, okay? So he called that process transformation. So the transformation happens to the R strain into S strain. So again, the first part is the S strain. He injected the S strain directly in a mouse, and then the mouse dead. And the other part, he injected the harmless bacteria, which is the R strain, in the mouse, and the mouse still alive. The second part of the experiment were done by heat killing the bacteria, which is the S strain, and once he injected the heat killed bacteria inside the mouse stay alive. And the third part, he make a mixture between the harmless bacteria and heat killed bacteria, or heat killed S strain, and he injected the mixture into the mouse, and surprisingly the mouse dead. And when he took a sample from the death mouse, from the lungs of the death mouse, he grew it and he found that the bacteria that is found in the plate is S strain, live S strain, or live disease causing bacteria. So he called this process as a transformation, because one type of bacteria had been changed permanently into another. So R strain here is changed into S strain. So because of the ability to cause disease, he was inherited by the offspring of the transformed bacteria. So he concluded that the transforming factor had to be a gene. So there is a gene inside the heat-killed bacteria that changed the R bacteria. So this is the first experiment were done by Griffith. The other experiment, which is the molecular cause of transformation, were done by Avery. So Avery, he extracted a mixture of various molecules from the heat-killed bacteria. So he took the heat-killed bacteria, okay, the S strain, the heat-killed S strain, and treated it with mixture of enzymes, okay? By this point, you need to know that what's the main enzyme that's used, and for each enzyme, what is used for. So he extracted many molecules like carbs, proteins, RNA, DNA, and other molecules. And he uses enzymes to kill or to destroy all of these molecules, okay? Once he used the enzymes to destroy all of these molecules, he concluded that the carbs is not the genetic material. At the same time, when he used another molecules or enzyme to destroy the proteins, he also concluded that the protein is not the genetic material. And after that, he also used another enzyme to destroy the RNA, and he concluded that the RNA is not the genetic material. And after that, once he used an enzymes to destroy the DNA, he found that the DNA is the genetic material. So again, this experiments were done on the extracted mixture from the S strain, which is heat-killed S strain. The mixture contains many molecules: carbohydrates, proteins, RNA, DNA, lipids, and so on. And he used many enzymes. Each of these enzymes is used by step by step. In the first time, he used enzyme that destroy the carbs. In the second time, he used the enzyme that destroy the protein. In the other time, he used the enzymes that destroy the RNA and the lipids, and then the DNA. Once he destroyed the DNA, he discovered that the transformation didn't happen. So the main cause for the transformation or bacterial transformation were the DNA. So the transformation happens when the DNA is found. Once the DNA is not found, the transformation will not happen. Okay, is it clear? So again, the experiment, once they destroy the DNA in the mixture, the transformation didn't occur. So the transforming factor is the DNA. So by observing bacterial transformation, Avery and other scientists discovered that the nucleic acid DNA stores and transmits genetic information from one generation of bacteria to the next. So these experiments were done to check what is the chemical nature of the genes or genetic material. So by Griffith experiments and Avery experiment, we discovered what is the genetic material. Okay? Let's check the last experiment, which were done on the bacteriophage. This is the bacteriophage, T4 bacteriophage. The bacteriophage, which is the bacteria eater, which is a specific virus or a kind of virus that will infect the bacteria. So bacteriophage is a virus, by the way, okay? So make sure that you will understand this point. Bacteriophage is a virus that will infect the bacteria. The structure of the bacteriophage, like this. The DNA, which is here on the coat, and inside the DNA, again, this is the DNA, and we have a protein coat. Again, this is the DNA in the middle, and coat, tail fiber, tail sheath, all of it is made by protein. So the internal material is the DNA, and the outside material is protein. So the bacteriophage is made up of protein and DNA, just protein and DNA. So the experiments were done by using radiolabeled material like phosphorus 32 and sulfur 35. By the way, he chose these two radiolabeled or radioactive isotopes because the phosphorus is the main, which is one of the things that is found already in the DNA. And at the same time, sulfur is found just in the protein. So the sulfur is not found in the DNA, and the phosphorus is not found in the protein. So another scientist called Hershey and Chase, they studied the bacteriophage, and they want to check if the DNA or protein is the genetic material. They want just to make sure that the DNA is the genetic material. They need to approve, either to approve or decay the theories done by Avery, okay? So they wanted to determine which part of the virus, either the protein coat or the DNA core, will enter the bacterial cell. So in the first part of the experiment, they labeled the bacteriophage with radioactive isotope P32, which is phosphorus 32. Once they labeled the bacteriophage with it, the DNA that is found in the bacteriophage here is the DNA that contains the isotope here, the phosphorus 32. And the other part of the experiment were done by bacteriophage, which is labeled by sulfur 35 in the protein coat. So the sulfur 35 will be found in the coat. Look at the small dots here, which is the radiolabeling, okay? Or radioactive isotopes, okay? Once the bacteriophage with phosphorus 32 inject the bacteria, they will insert specific material. They want to check what is the material that will be injected. Is it the DNA or is it the protein? So by this point, they found, or by this experiment, they found that the bacteriophage with phosphorus 32, it will insert the DNA inside the bacteria, while the labeled DNA inside the bacteria. While here, the bacteriophage with sulfur 35, that's already found in the coat, will not insert any radioactive material inside the bacteria. And after a screening for the infected bacteria here and infected bacteria here, they discovered that the genetic material is surely the DNA. So the DNA with radioactive material is found inside the first one, which is infected already with the bacteriophage with phosphorus 32. So the role of the DNA, they want to know what is the role of the DNA. The DNA that makes up genes must be capable of storing, copying, and transmitting the genetic information. So the function, storing, copying, and transmitting the genetic information in the cell. And that is the end of the lecture. Have a nice day, and thank you. I want you, miss, to make sure that you will understand the points, and please watch the video to know what's the main